Today's date is January 15th, 2021. It's the Friday before a long 3-day weekend, so I know we're all excited about that. We're half of the way there. You've got this block and next block, and then you're in freedom, I suppose. But let's go ahead and make sure we work hard and take some solid notes, starting with this slide. So the title of today's lesson is Enzymes. It's Enzymes Part 1, to be specific. We've got 2 objectives and an essential question today. The first objective is for biologists to be able to explain how enzymes act as catalysts for biological reactions. The second objective is for biologists to be able to explain how an environment can impact an enzyme's ability to function. We won't talk much about that today. That's really going to come up as part of tomorrow's or Tuesday's conversation. But I just wanted to give you all that idea, that thought, so you can start to toss it around in your heads. The essential question, this is our guiding light. What do we want to be able to answer by the end of this lesson? What does it mean for enzymes to have an optimal environment? I think you guys will have some ideas about the answer to that question by the end of today, but it will really come on Tuesday. So this is Unit 1, Day 4. When we're taking strong, effective, efficient, cohesive, and consistent notes, we're taking notes that start off with a title and a date. That way we can return to our notes and see, this is exactly when I covered this, and this is exactly what I covered in this lesson. So make it easier on yourself. Make sure you've written down at least the title and the date, and perhaps even the objective if you want to go above and beyond. All right, so for the last couple of days, over our 4 biological compounds. And as I have explained to you all in the past, what I really want you to know is 4 things about each of our 4 biological molecules, which is why we've got 16 boxes here, 4 by 4, 4 times 4 is 16. So for each biological compound, I want you to know the monomer that makes up that compound, the elements that make up that compound, the function of that compound. So what does it actually do for a living organism? And then I want you to know some examples, common examples. So we knocked out the first 2 rows of this table yesterday, the rows about carbohydrates and lipids. And then we introduced nucleic acids and proteins during yesterday's lesson as well. So we've got to knock out those two rows today. Hopefully you all have already copied down this table in your notes, and you've already written down the information in the first 8 boxes. And now it's just a matter of adding on those 2 rows at the bottom to knock out the second 8 boxes. But let's go ahead and quickly move through these first 8. So under box 1, what's the monomer for a carbohydrate?
Glucose.
Glucose. And there's another word for it. What's that other word? Good, thank you.
Monosaccharides.
Abigail. Yep. Oops, thought I was over here. Monosaccharide. And what 3 letters typically indicate that we're talking about a sugar?
The O's.
Excellent. Thank you, JT. Thank you, Sally, and thank you, Melissa. We should be looking for OSE. Excellent. What elements make up a carbohydrate?
CHO.
Carbon, hydrogen, and oxygen, known by their abbreviations C, H, and O. And what's really special about those 3 elements as it relates to carbohydrates? Good, thank you, Sally. You anticipated what I was going to ask. They appear in a 1:2:1 ratio. What's the function of carbohydrates? What do they do for living organisms?
Producing quick energy.
Excellent. Thank you, Abigail, and thank you, JT. They provide quick energy, and we'll talk about it in Unit 2, but they also provide structure in the cell walls of plant cells. And then lastly, can we think of some examples? I want to get as many examples as we can think of for carbohydrates.
Sucrose.
Sucrose is one. What about some folks who I haven't heard from? Janelle and Tyshawn and Santuan and Sienna.
Starch.
Good, thank you. Starch is one. Lisa, did you have something?
Glycine. I forgot how to say it. Glycogen.
Yeah, glycogen. Thank you, Lisa. And then I heard Josue said something, fructose. I think you said fructose, Josue. So I appreciate it. All right, so those are good examples. And again, anytime we see OSE, we should be thinking about a sugar. Excellent. All right, let's move on to the second row, lipids. What's the monomer that makes up a lipid?
Glycerol and 3 fatty acids.
Excellent. Glycerol and 3 fatty acid tails. So it ends up getting shaped like a capital E. What about elements? What elements make up lipids? This is an easy one.
CHO.
CHO, excuse me. Same elements, but they do not appear in that ratio. Okay, so we've got these same 3 elements, carbon, hydrogen, and oxygen, appearing in all 4 of our biological compounds. But as we will see in the next 2, in nucleic acids and in proteins, we have some additional elements as well. What do lipids do for living organisms? What do they provide?
Long-term energy.
Good, Lisa. Long-term energy. And is there anything else we can think of?
Insulation.
Insulation. Thank you, Abigail. It's not there, but you are 100% right. In Unit 2, and that we'll come back to that answer in the next slide. In Unit 2, we will also talk about how lipids are an absolutely essential part of the cell membrane. In fact, they are the majority of the cell membrane. Lastly, box number 8. What are some examples of lipids?
Waxes and steroids.
Waxes, steroids. Can we think of any others?
Oils and fats.
Oils and fats. And then phospholipids, that's a bigger word, but I think you probably can see the 2 words that are combined to make that word, phosphorus and lipids. We'll talk about that in Unit 2 as well. Okay, so this is information that we ideally already had written down. We did reveal these 2 rows yesterday, and then also yesterday we introduced the next 2 rows. So let's see how well we can do with nucleic acids. Under box number 9, what's the monomer of a nucleic acid? Nucleotides. Beautiful. Thank you, Abigail. And nucleotides have 3 parts. They have a 5-carbon sugar, which we could call pentose. They have a phosphate group and also a nitrogen base. Okay, sorry about that. I needed to answer a quick phone call from a parent. What are the elements that make up a nucleic acid?
Chomp.
Good. Chomp. I never thought about it like that, but Melissa was ready to go. She had typed them all out. She is correct. So chomp, that's nice. So carbon, hydrogen, oxygen, nitrogen, and phosphorus. CHONP. What is the function of nucleic acids? What does it do for living organisms? What do they do for living organisms? Good. They carry the genetic information. And then they also, as we'll find out again in Unit 2, control all of the cellular activities. They're the bosses of the cell. They tell the cell exactly what to do, when to do it, and what to make, and how to make it. Lastly, what are our two examples of nucleic acids?
DNA and RNA.
Thank you, Dejan. Yep, DNA and RNA. Okay, last row. So what's the monomer of a protein? I'll give you a hint. There are 20 of them. 20.
Amino acids.
Yes, good. Amino acids are the monomers that make up proteins. And what elements are present in proteins?
Carbon, hydrogen, and oxygen, nitrogen.
Excellent. Carbon, hydrogen, oxygen, and nitrogen. So again, we've got those same three present in all four biocompounds: carbon, hydrogen, and oxygen. What is the function of proteins? What do they do for living organisms? That's kind of a trick question, so I'll reveal this myself, because they kind of serve as the do-it-all compounds. Most of what makes living things living are proteins. The vast majority of the biocompounds that you would find in a living organism are proteins. So they do almost everything besides these things. And then can we think of some examples? We did have an asynchronous assignment yesterday in which I asked you all to do some brief research about 5 types of proteins. Do we remember what those 5 proteins were? Insulin.
Hormones and antibodies.
Good. I heard hemoglobin, antibodies, insulin, and then there were 2 others. One that we'll talk a lot more about today called enzymes. And then which one did I skip over? Which one did I not know? Oh, hormones. Hormones. Good. All right. So I like this table a lot. I love visual representation. If you're like, this is too geometric for me, this is really basic and ugly, we're going to take a look at another visual representation, a flowchart or a mind map that might be a little better suited to my abstract minds in this class. But I love this table. I would encourage you to take a picture of it. Start to make some connections. Again, yesterday we briefly talked about the connection between nucleic acids and proteins. See if you can think of any other connections and push yourself to do that. It'll really pay out dividends in your test performance. So I'll leave this up for about 60 more seconds. Okay. So this is what I was talking about, the mind map that you all might consider including. So it just provides a different look at things, another way to represent this information. You should, based on the information that we have in our notes at this point, be able to answer and fill in this chart. You should be able to. If you can't, then that means you do have more work to do. You should go back and look at our previously recorded live sessions from Wednesday and Thursday of this week. And you should also make sure that you're paying close attention to the notes that you're taking going forward. All right, we really have to emphasize this first unit is so foundational, and it means such a great deal for the course that you all understand this. Okay. So here it is. Here's the information. Let's see if we can fill it out. So as you guys can see, in this mind map, some information is given to us. And we've got to use that information to fill in the blanks. Of course, we've got our 4 organic compounds that will appear up here. So in this first stream, for example, we're told that blank are made of amino acids. So what information should be in this oval?
Proteins.
Proteins, yeah. So proteins are made of amino acids. And examples include what? We've got antibodies. What were the others that we just talked about on the last 2 slides ago?
Insulin and hormones.
Insulin and hormones. And then we had 2 others as well. Does anybody remember? One that we're really going to talk a lot about today is the title of today's lesson.
Enzymes.
Enzymes, and hemoglobin is the last one. So of course there are hundreds of thousands of different types of proteins. There are proteins all over the place. But in this class, we really don't talk specifically about any besides this 5. So besides those 5. So make sure you have those examples written down. Okay, the next stream. We're not given much information here. All we're told is that blank, which are made up of blank, and this blank, the second one, can also be known as cellulose. So which organic compound are we talking about here? If we see cellulose. Yeah, Abigail, you're right. Thank you. I'm sorry, I cut you off a little bit. Carbohydrates, I'm pretty sure that's what I heard you say. So what are carbs made of? What are carbs made of? Sugar, there's a better word for it, more scientific word for it.
Glucose.
Glucose. Glucose is actually going to be this circle right here. So there's another phrase that we can use. Sally is definitely on the right track. Thank you, Sally. Excellent. Monosaccharides. It's a difficult word to say, maybe. Monosaccharide. And then Lisa just said that monosaccharides are also known as glucose. Okay, and in this third stream, we are told that we're talking about lipids. What are lipids made of?
Glycerol.
Glycerol and 3 fatty acids. Can't forget the fatty acids part. I know it's kind of like, oh yeah, I forgot to include that. But those 3 fatty acids are really important because they give the chemical properties to the lipid. They determine the lipid's chemical properties. So we can't forget those 3 fatty acids. What are the jobs of lipids? What do lipids do?
Long-term.
Thank you, Janelle. Insulation, and then long-term energy. Dijon gave us that one as well. Good. So again, I love mind maps like this. We got one more stream, but if this is something that you like this image and it helps you to organize things specifically, you could copy this down and add on to it. Make this stream longer or have a stream for examples. Just gives you another way of looking at the same information. In our last stream, we're told that the two main types of this organic compound are RNA and something else. So RNA, which organic compound is that?
Nucleic acids.
Good, thank you, Abigail. Nucleic acids, which are made of what? What's the monomer of a nucleic acid? Or the building block or the subunit? All mean the same thing. Good. A nucleotide. Thank you, Melissa. If RNA is one of the main types, then the other main type must be DNA. Thank you, Janelle. And that ultimately will lead to genes and then proteins. All right, so I'm going to start this timer, 2 minutes. You can do whatever you want to get this information down over these next 2 minutes. Just take a screenshot, take a picture, write it down. For some reason, I think it's because my internet is slow. You can see that the video is not actually moving second by second. So this is going to be a long 2 minutes. Okay, that took a long time. That was more than 2 minutes, but hopefully you got it down. Let's move on. So At the end, we talked about the fact that nucleic acids eventually lead to proteins, and genes are really the connection in that process. A gene is a small segment of DNA that codes for at least one protein, but often more than one protein. Let's call it between 1 and 5 proteins. So, for example, each of us is made up of unique DNA. The cells in our body have unique DNA. Human DNA is about 3 billion base pairs long. So just imagine if I were to sequence my DNA, I would have a book that would have 3 billion letters in it. It would be a whole bunch of A's and T's and C's and G's, and they'd be in different orders: A, T, 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 C, C, G, A, T, A, C, G, A, T, C, A, so on and so forth, for 3 billion letters. But only small portions of that book that's made up of 3 billion letters, only small portions of it would actually be genes, things that actually code for proteins. So human DNA, again, has 3 billion base pairs, but there are only about 30,000 genes in human DNA. Still, that's a lot because they sometimes will code for more than one protein. But it's not the entire DNA that gets used in every cell. So for example, the cells that are in the back of your eyes that allow you to process light, those might have genes 1, 2, and 3 activated. And then the cells that are in the back of your eye that allow you to process depth, how far something is away from you, those might have genes 1, 4, and 5 activated. And then the genes that are in the muscle that surrounds your heart, those might have genes 3001, 3008, and 4005 activated. So it's not that every cell in your body has every gene activated. Every cell only needs to have a certain set of genes activated in order for it to do its job. And this is a remarkable and beautiful process because it allows cells to specialize. So the cells that are in your eye are not the same cells that are in your stomach, which are not the same cells that are in your bones, which are not the same cells that are in your spinal cord. They all do different things. They're shaped just slightly differently. They all have the same DNA. They all have that book of 3 billion letters, but they go through and they choose which sections of that 3 billion are they actually going to use? Which sections are they going to turn on and read? We'll talk a lot more about that, but genes are really, really special. And so we've got to differentiate. Genes are not DNA. Genes are sections of DNA. And here's the big idea, though. The nucleotides that make up DNA, the A's and the C's and the T's and the G's, those eventually get converted through a process called transcription to RNA. And we'll talk more, we're going to dedicate a whole unit to this. RNA is then going to signal the cell to say, okay, so I got this code. My code is AUG. So I need you to bring this amino acid. And then the next section of RNA will say, well, my code is GUA. So I need you to bring this amino acid. So of the 20 amino acids, they all have different combinations of sequences that signal them. So CCA signals for a specific amino acid. CCG might signal for a different amino acid. AGG signals for a different amino acid. And eventually, once the entire signal has been read and all of the amino acids have been triggered to come into place, now you've created a chain of amino acids that will become a protein. So AUG is typically first. AUG brings its amino acid, And then maybe we have CCG. It's going to bring its amino acid, and those two get linked. And then we've got GUA. It brings its amino acids, and now you've got a chain of 3 called the polypeptide chain. It grows and grows and grows and might be 300 amino acids long, and then it gets cut off, and then it'll fold and become a protein. And this is the process of protein synthesis. So we can see how we went from nucleotides, A's, T's, C's, and G's. Those get converted to RNA, to A, U, C's, and G's. Those bring in amino acids, and then those combine to form proteins. It's a fluid process, and it's how we relate DNA and proteins. You do not need to write this down right now. This is going to come up a lot later, but we need to expose you to this idea early on. There is a relationship between the nucleotides that eventually become a nucleic acid and then the amino acids that eventually become a protein. This is the general sequence: nucleotides, DNA, amino acids, proteins. And along the way, again, we keep coming back to our central theme, which is that structure determines function. So if my order of amino acids is even slightly off, it can totally change the protein that I make. Yesterday, I talked about my recipe book analogy. If my order is off, if I put the egg on the skillet before I actually mix in the salt and pepper, the end result is going to be different. Or if I mix in the vegetables before I saute them on the skillet, the end result is going to be different. So we need to follow a specific order. Otherwise, the protein that we make could be mutated. It could lead to very serious cancers, very serious diseases. Okay. So the protein that we're talking about today, and this is what I do want you guys to write down, the stuff especially that's highlighted in yellow. Enzymes. Enzymes are a specific class of proteins that carry out various functions in living organisms. The primary function of enzymes is to serve as a catalyst. We can see that word here. It's highlighted, bolded, and underlined. Catalysts increase the rate of a reaction. They speed up a reaction. So an enzyme's primary job is to speed up a chemical reaction. That doesn't mean that the chemical reaction needs an enzyme to take place. Most chemical reactions will happen on their own spontaneously if given enough time. But just imagine how dangerous that would be for the human body. If we had no enzymes, that would mean that even the signals in your brain would not travel as quickly as they do. That would mean that your food would not be digested as quickly as it is. That would mean that your muscles wouldn't be able to contract as quickly as they do. So we need enzymes to carry out our chemical reactions at a much faster rate. And every enzyme, every type of enzyme has a unique shape. And that unique shape is determined, again, by the sequence of its amino acids and based on how it folds. We can see an enzyme folding here. It's folding into place. It starts off kind of like a shoelace that's been stretched out. It's just a chain of amino acids. And then we're going to kind of tie that shoelace up together in a really specific and intricate way. We can see in this 3D rendering would look like small spherical balls. Those represent the amino acids. So again, they start off as a polypeptide chain. They're just amino acids that have been linked together. But based on the chemical properties of those amino acids, the enzyme is going to fold. So I talked about it yesterday, but some enzymes like water. So that means that they want to face outwards. so that they can be exposed to water. Some enzymes hate water. In fact, they cannot tolerate being around water. That means that they need to be put on the inside of the enzyme to be insulated, such that they never come into contact with water. Some enzymes are positively charged. So they really, really like to be near negatively charged, not enzymes, but some amino acids are positively charged, which means that they really like to be near negatively charged amino acids. Some enzymes like to form specific bonds with other, I keep saying enzymes. Some amino acids like to form a specific bond with other amino acids. So they have to be near them to do that. And so all of these things, all of these chemical properties coming into play force the enzyme to fold in a really intricate way, giving it its unique shape. So this is why you would see, if I mess up and I put in the wrong amino acid, that could be a big deal. If I put in an amino acid that doesn't like to be around water, where I'm supposed to have one that does like to be around water, then I totally change the shape of the protein. Because now I've got something that needs to be insulated where there should be something that wants to be facing outward. So I'm totally changing the shape of the protein in that case. Even if my protein is made up of 2,000 amino acids, even a small mistake like that can have big, big implications. For example, some of us may have people in our lives who have dementia or Alzheimer's disease. At this point, and for basically the last 20 years, we've been able to sequence the entire human genome. And so we know that the mutation that leads to early onset dementia is caused by one very, very small mistake, one issue, one error in the human genome. And it leads to a messed up protein. It messes up only one amino acid, which then messes up the entire protein. It prevents the protein from doing what it's supposed to do. In this case, it leads to small blockages and the buildup of a specific type of plaque in your brain, which can slow down the way that the messages are communicated. Okay, so small issues, and this is what's so remarkable, because that doesn't always happen. You know, the human body is really, really good. It has all these checkpoints in place to make sure that that error does not ever come up. And that's something we'll talk more about. So enzymes have several really cool properties. One is that they are specific, which means that they can only act on substrates that have a specific shape. So now you're like, what is a substrate? Well, we've got that defined on the screen as well. A substrate is a reactant in a chemical reaction. So this means that it is what's actually being acted upon. The reactant is being acted upon. It might be broken down. It might be combined with something else to build something, to synthesize something new. But the substrate is the object that is being acted upon. And where does that action take place? Well, it takes place fittingly in the active site. The active site is the site of the action. It's where the chemical reaction actually takes place. It's where the substrate is inserted and it's going to come out slightly different. It might come out bigger. It might come out smaller. It might be totally destroyed. It might be combined to create something new. But whatever that chemical reaction is doing, it's going to take place in this active site. Now, see in this image, we can see how they fit perfectly together. They're like it's like a slice of pie being reinserted into the whole thing, or a puzzle piece, or a lock and key. There are several different analogies that work here. So for example, the keys that allow me, there's only one key that allows me to get into my apartment is not the same key that would let me into your bedroom or into your house or into even my classroom at West Meck. It wouldn't even let me into my own car. We've got different keys for different locks, just like we need different enzymes for different substrates. And the way the key works is based on its shape. What are the teeth of the key shaped like? That's ultimately going to decide how it works. Same thing applies to enzymes. The shape matters, and that's what we call specific or specificity. So let's take a look at this. Question, which of the following enzymes is able to act on hold on, bear with me. Don't look at the answer. Don't look at the answer. Okay, which of the following enzymes is able to act on substrate G below? So there's substrate G. We've got to take note of how it is shaped. So which enzyme is going to fit with substrate G?
Number one.
Yeah, number one.
Number one, we've got everybody chiming in, everybody saying enzyme 1. You are all incorrect. No, I'm just kidding. You're all right. So yes, we can see how the shape of substrate G fits perfectly in the active site of enzyme 1. Let's go back. So we can see the active site here. That active site has to be shaped perfectly to allow substrate G to fit into it. In this case, in enzyme 2, we've got an active site that is shaped like 1/2 of an arrow. So that's not going to work. They don't fit together. Enzyme 2 would not be able to do anything to substrate G. Enzyme 3's got an active site that is shaped like some type of wave or flag. Again, not going to work. Okay, so the specificity is a factor of the shape of the enzyme. All right, so I want you guys to copy down this diagram as well. I'm going to start this timer for another 2 minutes. Copy this down. Make sure that you're labeling things, the same things that we've labeled here. So everything in this purple box, you don't need to include this word H2O. We're not going to talk about that, but everything else I think you should try to write down. Okay, so the image is going to be on the next slide. But I want you to take note of something. Number one, we've got this enzyme, and I can guarantee you this question will show up, not question, but not this exact image, but a very similar diagram will be on almost all of your tests, probably including the EOC exam this year. So if you can know what's present in this diagram, and if you can identify different parts of this diagram, you are in a good place going forward. We can see the active site. It looks like kind of like a hole in this case, one side that's shaped like a hexagon and one side that's shaped for a pentagon. We can see the reactants, AKA the substrates, being moved into the active site of the enzyme. The active site is where the action happens. So we can see that in this case, the enzyme is breaking down the substrate. It's breaking down what started off as one thing and separating it into two unique things. But what I really want you all to notice here is that the enzyme starts off exactly how it ends up. It is unchanged from beginning to end. It's not affected by the chemical reaction. It maintains its shape. Okay, so this is important because what it means is that enzymes are reusable. They can undergo the same reaction time and time again. Now, eventually they will be deactivated, essentially. They might be broken apart and repurposed, but they can undergo the same reaction several times. This provides a great benefit to the cell. So that's the other quality of enzymes that I want us to be aware of. They're both specific and they are reusable. They're unchanged in the chemical reaction and can undergo that same reaction several times. So it breaks apart this substrate, and then it might go and find another substrate and break that apart. And then it might go and find another substrate and break that apart. And we're going and doing the same thing over and over again, and we're doing it quickly. Remember, enzymes are catalysts as well. They speed up the chemical reaction. Okay. So in review, enzymes are three things, and I just said them. Number one, they are catalysts. They speed up the chemical reaction. This chemical reaction would almost definitely still take place by itself without the help of an enzyme, but it would be slow. So we use enzymes to speed things up. Number two, enzymes are specific. They work with unique substrates. So not everything is going to work with every enzyme. Enzymes have very specific substrates that they work with. And number three, enzymes are reusable. So they remain unchanged throughout the reaction. That's what RXN means here at the bottom, reaction. So those are three qualities we've got to keep in mind about enzymes. And we'll learn more about enzymes and some other cool properties that they have in our next lesson on Tuesday. But we've got 15 minutes now. And what I want you guys to do in these 15 minutes, one of three things. The exit ticket, which I would like everybody to do. Number one, if you have completed all of your assignments for this week, then you can get ahead. Even though we're on day 4, you can go ahead and start U1 day 5, Unit 1 day 5, the Amoeba Sisters enzyme video and quiz. But if you know that you missed a couple assignments, a warm up, an exit ticket, or one of the asynchronous assignments this week, or even last week with Unit 0, then you can take the next 15 minutes to work on those. All right, so we've got a little bit of choice incorporation here. I will be on mute, but I will be available to answer any questions you have, so just let me know. Okay, folks, so it is now 1:20, and I hope you all have had a great third block, and I hope you have an even better fourth block to close out your day. I will talk to you all on Tuesday. If you need anything between now and then, do not hesitate to let me know, and I hope you have an amazing weekend.
You too. See you
Thank
soon.
you.